Hello, my name is Laila Kudsiova. I'm a senior lecturer in pharmaceutics, and I would like to give you a very quick overview of my research interests and expertise in the field of gene therapy. The aim of gene therapy is to introduce therapeutic DNA into the cell nucleus to produce a protein that can then treat or cure a disease. But in order to deliver the DNA all the way to the cell nucleus, we need to have an effective gene delivery vector that can overcome numerous cellular barriers. So my research interests lie in the discovery, optimization, and characterization of non-viral gene delivery vectors using novel formulations composed of things like lipids, peptides, or polymers. The vectors that I have studied most extensively are lipoplexes, which are prepared by mixing cationic liposomes with DNA, and when characterized by techniques such as dynamic light scattering, electron microscopy, and small-angle neutron scattering, we found their structure to be very ordered, with DNA sandwiched between lipid bilayers. This structure, however, changed drastically when a cationic targeting peptide was introduced into the formulation, now producing lipopolyplexes, resulting in much more condensed solid nanoparticles. In vitro tests in breast cancer cells showed that lipopolyplexes were much more effective than lipoplexes at transfection, possibly due to the vector's better ability to overcome cellular barriers and delivering the DNA into the cell nucleus, as shown by the red DNA fluorescence in the confocal microscopy imaging studies. We also tested similar formulations to deliver siRNA, which inhibits gene expression, and we found similar features to those of DNA complexes. However, when we used circular dichroism, which is a spectroscopic technique that can be used to determine the secondary structure of large molecules like DNA and proteins, we found major structural differences between DNA and siRNA complexes. For example, DNA conformation changed. It became more twisted, with increasing lipid concentration in lipoplexes, which we did not observe in the more rigid siRNA formulations. And in lipopolyplexes, the peptide preferentially changed its conformation more drastically to bind to the siRNA than to the DNA. In another study, we have synthesized a series of tri-chain lipids, and we found them to be much more effective in transfection than their corresponding di-chain lipids. And when we studied their structure by small-angle neutron scattering, we actually found that the tri-chain lipids produced a much thicker bilayer as a result of the third chain bending over or protruding into an adjacent bilayer, causing interdigitation, and we think that this is favorable for transfection efficiency. We also used giant vesicles to study interactions between vectors and model endosomal membranes. For example, in this experiment here, we can see dye release from a giant vesicle due to the destabilization caused by the addition of lipoplexes. The giant vesicle study actually led to the discovery of a novel fusogenic helper lipid, which was shown to improve the transfection efficiency of both lipoplex and lipopolyplex vectors by up to 7- and 4-fold respectively. And we think this is due to better fusion of the vector with the endosomal membrane, causing more DNA to be released from the endosome and access the cell nucleus, as shown here by confocal fluorescence imaging. So thank you very much. That was just a very quick overview. Future studies will look at synthesizing further structural analogs of the novel helper lipid and other lipids in order to further optimize and discover novel, more effective gene delivery vectors in the future.